Right, so welcome back to the second part of Galaxy. I hope you all can hear me. Can you hear me, class?
Yes, sir.
Okay, good. So let's continue from where we had left the last time. And there were a few questions yesterday. We'll answer those, and then we'll continue with our process of using Galaxy to identify the, bacterial resistance genes and antimicrobial resistance genes in bacteria, and also assemble the long read fragments into a actual assembly. So let's get started. Let me share my screen first, and then of course we'll start again. And here you are. So it's a bit humid here today in Chandigarh.
Sir, excuse me, sir.
Yeah.
Sir, I was able to install my Galaxy, but in Windows, not in Linux. There I was occurring some problems.
You're able to install your Galaxy in?
Windows, sir.
So which is fine. This is so you did a local installation, you mean?
Sir, actually, ORG site is not supported in my Linux. That's why I was not able to access that.
I see. Okay. So for now, you do it online, and then we'll see how we can go about it, right? So here we are.
Excuse me, sir. Sir, I was able to run the nano plot actually.
Yeah.
I got 8 plots for one
Yes.
data, and the 1 6 plot only I got. After that, I was not able to proceed actually. Filled log and all was there. So I got
Sorry?
only those 8 plots. After that, I was not able to continue due to some
So
error.
that happens. I told you there is a lot of queuing that happens in Galaxy. So it takes a lot of time to actually proceed from one step to the next step. Sometimes, depending on how busy the server is, how what is the condition of the server, sometimes it takes up a lot of time. So
Yes,
did
sir.
you not submit the next job, or you just got stuck
Sir,
at
I
this
got an error. I got an
error?
error.
Okay,
Yes,
so
sir.
there must be some problem with the input. We'll come to it
Yes,
and
sir.
we'll see. Okay.
Yes, sir. Thank you.
So here we are. We are doing NGS data analysis with Galaxy, and if you look at the steps here, so sequence data is coming from sequencers, so just raw data. And to derive meaningful inference, information from raw data must be processed. So you first do a quality control, then you assemble it together, then you finally go for annotation. So these are the steps that we're going to follow today. And then a whole suite of tools are available, both command line and in graphic user interface to assist NGS data analysis. And Galaxy is one such tool that allows you to do it online, or you can also do a local installation of Galaxy in your local machine and use it. And this is basically a web-based platform for data-intensive biomedical research. And this is basically for those who do not know hard code programming, but they can still do NGS data analysis using Galaxy. Galaxy is an open source platform, follows FAIR principles. We've talked of this yesterday. Findable, accessible, interoperable, and reusable is what is the FAIR principles. And this is the general principle that is now being followed for data policy across the world for all researchers, right? Then it uses tools from various domains that can be plugged into workflows and through its graphical web interface or GUI. And basically, the workflow can be shared. So somebody else can use the same workflow as yours, input his data from the one end of the pipeline and get the final process data from the other end of the pipeline. So that is what Galaxy facilitates. And then, of course, it manages data by sharing and publishing results, workflows, and visualizations, ensures reproducibility by capturing necessary information to repeat and understand data analysis. And Galaxy community is actively involved in helping the ecosystem improve and sharing scientific discoveries across the world. So you have a lot of people who are available online to help you out in case you get stuck at a specific point in your analysis, right? So this is the paper that we are going to refer to. What this has is complete genome sequences of 8 methicillin-resistant Staphylococcus aureus. So this is what you came to know when you sequenced it. So earlier what you had was a sequence of Staphylococcus aureus, and then you assemble it together, checked for genes for antimicrobial resistance, and you found them. So this is basically the final derivation of the paper, that you have complete genome sequences of 8 methicillin-resistant Staphylococcus aureus strains isolated from patients in Japan, right? So Staphylococcus aureus is gram-positive or gram-negative? Anyone? Staphylococcus aureus, gram-positive or gram-negative? Huh?
Negative.
I think it's gram-positive. Yes, it's gram-positive. This is a Staphylococcus aureus, right? This is gram-positive. And we call it MRSA because this is the one we are talking of specifically is methicillin resistant. So this is basically a Staphylococcus aureus strain that is resistant to all beta-lactams that are available, right? So beta-lactams include your methicillin, penicillin, then amoxicillin, and oxacillin, and other types of such beta-lactams, right? So this is resistant to all of them. So that is where the problem is, and these resistant ones are sometimes also pathogenic. Usually the bacteria is not pathogenic, and around 25 to 30% of individuals in a population carry this bacteria on their skin and nose, but these are healthy people, not really affected. But sometimes it becomes pathogenic and causes severe complications, including nosocomial infections. Clinical manifestations include from asymptomatic colonization to nasal mucosa, to colonization of the nasal mucosa, and to soft tissue infection, to fulminant invasive disease. So you could have a full-blown disease, or normally it is not so pathogenic. But the resistant ones are a problem because these can move around and become more and cause a spread of disease, right? So this is what we have. Then at the end of the analysis, we'll have a workflow that can be shared and that can be taken forward. And it could be, if you have another sequence data that you want to analyze through this pipeline, you just have to feed it at the first step, and all the steps will be repeated. And then finally, you'll have the results at the end, right? So this is what we have. What are we going to answer? We are going to answer some questions here. How to take the quality of MinION data. MinION is nanopore sequences. How to perform an assembly with MinION data. What species do I have and what is its sequence type? Which resistance genes are present in the genome that I'm currently assembling? And what are the genes that are present in my genome? So this is basically when you do the annotation, you'll have this, right? Then the tools used. So first, of course, for data upload, we're using paste and fetch. We have a URL from Zenodo that we'll use to upload data. I'm sure all of you have done this step. Then you uncompress the data. So there are several tools, depending on what is available. We'll use the tool that can basically give a file from BZ2 format to XYZ. So XYZ.bz2 is the compressed format, and then when you uncompress, you leave the extension, so it becomes XYZ, right? Then for filtering the data, we'll use filter long because we're having long reads. So we'll use filter long to filter the data, and we'll specify that minimum this length is what we want. Anything below this length has to be automatically dropped off, right? This is the first level of filter that we're doing. Then we'll do a comparison of filtered and unfiltered reads. So that will be done using nanoplot. And this is basically a data visualization tool. As somebody was mentioning that she got stuck here. So this is a data visualization tool that we'll use for comparing the filtered versus unfiltered data to see how our N50 values and other things have changed, right? Then we'll do the assembly. Assembly of filter reads will be done by Flye. So this is basically an assembler for long read sequences and specifically for nanopore. So we'll use that for assembly. Then to visualize how our contigs are formed and how does it look finally. So we'll use what is known as Bandage. So Bandage will give you a visual representation of your assembly. It will clearly show you that there is one large DNA and there is one small DNA, which is congruent to what we have in case of Staphylococcus aureus, where you have one chromosomal DNA and then you have one plasmid. So that would be basically just to kind of confirmation that what we are predicting is true. Also, the size of the assembly would tell you that it is a Staphylococcus aureus, right? Then you will assess the quality of the assembly using Quast. This is another tool for assessing the quality of the assembly. And then finally, you will check for the presence of AMR genes in your assembled data. So this will be done using Abricate. The original pipeline, the link that I've given you, they use different software, but for some reasons, it is not working. So we have used a slightly different software, but the end result is the same. Both are able to predict whether the assembly has your antimicrobial resistance genes or not. So known antimicrobial resistance genes are present here. So that will confirm that this is an MRSA strain versus a strain that is not having these genes. That would be a non-MRSA strain. You're following class, everyone?
Yes,
Class?
sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay. And then lastly, we'll do assembly annotation. So we'll find what are the genes, with what is the location of the gene, what is the individual, CDS and so on and so forth using pipeline again. This is called using a annotation tool called Procca. Procca is now the standard for prokaryotic annotation of genomes, right? So for annotation of prokaryotic genomes. So this is how we are going to go forward. And in this 1 hour, roughly, we'll have around now 45 minutes, so we will not have much time, but let's go forward. So what I've done is I've taken screenshots of everything so that it gets comfortable for you, and then I've also given you the help link that you can use for going back again and doing this. But before moving forward, let's answer some of the questions that were asked yesterday. So one of the questions that was asked yesterday was, what is the difference between the Fastq format and Fastq Sanger format? So the Fastq Sanger and Fastq format, basically they differ in terms of what is the ASCII characters that are used to represent your values, right? So this is basically, if you see the range of ASCII characters that you use in Fastq Sanger is 33 to 126, and it starts at 33, and then the ASCII characters from 30 to 126 are used for representation of Fred values from 0 to 93. I'll show you another slide where it will become clearer. And then if you look at early Illumina, it used 64 offset. So basically ASCII 64 was the first value to be used. And then from there on, it went on from 64 until the next one. And the range of the quality scores from -5 to 62. And then you have Fastq Illumina. Again, this is 64 base. So for all practical purposes now, ASCII base 33 is used. So this one, the top one, Fastq Sanger, is now the current standard. ASCII base 33, which is now almost universally used, and ASCII base 64, which is used in some older Illumina data, right? And to just, this is the paper from where I've taken this data. If anybody wants to see, you can go to this paper here. It is in Nucleic Acid Research 2010, and you know how to search for this paper, right? So you could take this down and you would be able to access the paper. And I really found it difficult to find this paper. It was not available so easily, in the sense that, to find the difference between Fastq and Fastq Sanger was really a task. But just to give you an idea of what we are talking about, so here is what is the base 33. So if you see here, a p error of 1.0, which means the quality score of 0 in Fred, basically the ASCII character that represents the 33rd ASCII character, which is the exclamation mark. So it starts at 33. The offset is 33, and it continues. Here it is given until 75. But as I've shown you, it goes a little further. If you see, go back here, it continues from 33 to 126. But largely, if you, I'm sure you know that largely we end up around 40. So 40 is 0.0001 error percentage. So that is a good quality enough. So we don't go beyond that for scoring. Sometimes you will find 41, 42, 43. So that is the probability of error if it is 41, 42. The higher the Q value, the lesser the probability of error, the more confident you are of your data, right? And this is the earlier scale. So if you compare here, the A here stands for a quality score of 32, Fred quality score of 32, which is very high score in the sense it is nearing 40. But in this case, if you use the 64 base, A stands for the quality score of 1, which is very low, and a high probability of error, 0.79 probability of error. You follow class, everyone? Right, so this is what you have. So for all your practical purposes, this is what we use. So Fastq Sanger uses this, and even Fastq Data uses this, and you can always interconvert between them. Everyone, you follow class?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
So this is the difference between Fastq and Fastq Sanger. And then, of course, we move on and we talk of Galaxy now. So I've already told you, when you look at the Galaxy interface, you will have 3 panels. The left panel is your tool shed. The right panel is your history, where as you move on and as you perform your experiments, you will have a whole lot of experiments showing up here. And the color coding is important. If it is showing in gray, it means it is queued. If it is showing in orange, it means it is being processed. If it is showing green, it means that the process is done and the process has been correctly done. If it is in pink color, then it means there is some error has happened because of the way you have inputted, because of the way you have input your data. So that color coding you will be able to see when you move on, right? So here you are. So this is your workbench. You can sequentially catalog the processes or the steps of the workflow here. And then, of course, this is your tool shed, where you can actually look at the tool that you want to use. So you can just go to search and type the tool name. It will come up on the top, and then you can click on it. That tool will now come in the center, and the inputs required for that tool would also be shown there, and you would be able to pick up the file that you want to input up here, right? So this is how you go about it. Then we move on, and of course you can name your history so that you you know what you're doing, and if you want to come back after, let's say, 2 days, 3 days time, you would still be able to, or let's say 2 months time or 2 years time, you still can go back to your history and you know exactly what you've done the last time when you were doing the analysis. So you name your history using, this is the pencil button. Pencil button is for editing. You can use this to rename your history and also to rename your files. Then this is where you can upload your data. You can click on upload data. There are several methods of uploading data. For lack of time, we'll just look at one, which is the most common one, where you're using your URLs to upload your files here. So this is where you go to paste and fetch. And once you go to paste and fetch, you click on it, you'll have this table here. You can paste your link the way we did yesterday. And you can see this is the Fastq VZ2. So this is a compressed format. Finally, when you start analysis, you'll have to uncompress your data, right? So therefore and then after that, you click on start. When you click on start, this will turn green, and that will indicate that the process of uploading the data has begun. It is still not uploaded completely because you see there is an orange indication here, and you can see there is buffering here, right? So the process of uploading data has begun, but it is not complete yet. Then when you actually get your data completed and the upload is complete, you can close this window directly after you have seen it green. You can close it and the process of uploading will continue. Once the uploading is done, this will turn green, right? So this is where your data is now finally uploaded and you can start with the next process. Then we move on and you can click on this panel. So this will give you some basic idea of the data here itself. And then of course you can click on this I button that will give you an idea of your data here. So your data will be, if it is a huge file, only part of the data will be shown. So you can see here, this data set is large and only the first few megabytes are shown here. If you want to see all of it, you can see show all. You can click on show all, and that will basically take some time to load, but over time it will show you everything. So here you can see this is our Fastq file very clearly. You have @ sign and the identifier in the first line. You also have the length mentioned in the first line itself. So this is 218. So this is not typically a long read sequence, right? So this is what we want to filter out before we start our analysis, because these can complicate our analysis later. Then you have the second line, that is the sequence line. Then you have the third line. I told you it will be a plus and then the repetition of the first line. So exactly the same thing. And then the last line is your ASCII characters, which represent your actual quality, Phred quality score. So you can go back to your ASCII tables and convert this into the actual Phred quality score in terms of 0 to 40 or 41, 42 as you wish, right? So this is how you do it. And then of course you have the next sequence here. So after the fourth line, you have the next sequence. So this one here is the next sequence I'm sorry. So this is in PowerPoint. So then again, you have the next sequence, 4 lines, then next sequence, 4 lines, and so on and so forth. So this is a huge file. And this is how you can have a view at the data. If you want to edit the attributes of the data, remember our file is compressed, so we need to decompress it. So we move on. And we can also have a look at data from this position here. If you click on this I button, this will also give you some properties of the data. You can have a look at that. Then, of course, you need to decompress. So first you click on this pencil, and that will give you this panel here where you can change the attributes. So in attributes, you can change the name if you want to. And then, of course, you want to decompress the file. So you go to the second tab and you say convert, right? So when you click on convert, this will give you some options. And then, of course, you can, select the option of what you want to convert it to. So this is, if you see, Fastq Sanger BZ2 to Fastq Sanger. So the .bz2 extension is gone, which means this is the uncompression that is happening here. So this is what we select. Sometimes you may not see this. For examples, in that case, you can take whatever option shows you or convert it from .bz2 to not .bz2. The extension is gone. That is the option that you're going to use, right? And those of you who are working currently and you're doing parallelly, you might not see this option of Fastq Sanger BZ2 to Fastq Sanger. You would see GTF BZ2 to GTF. So that option can be used, right? Everybody here with me? Class?
Yes, sir.
Okay, so because the time is less, we'll go a little fast, but of course, in case you get stuck, you let me know. And then, of course, I've also already shared the link with you. You practice at home, because if you do it online, it is going to take 5 or 6 hours to finish because of the speed and the data that we have is large, right? So let's move on. So once you've done the conversion, Then you come to, you say create data set. So now your conversion is done. You click on this. So you'll have this now coming up here, right? So this is now, if you see, Fastq Sanger BZ2 to Fastq Sanger on data 1. So this was your data 1. And now this process has been done on data 1. So it has now become Fastq Sanger instead of .bz2 compression. It is now an uncompressed file, right? So here you are, you create that. And then, of course, this becomes green over time and basically done. Then you could also tag your data here. You could write, so this symbol would be there with your data. You can tag it and use the hashtag also in the in front. I've not used it here. You can use hash or your pound sign and then the whatever name you want to give. So because this is uncompressed, you can write uncompressed. And here also, because the name was too large earlier, you could again go to attributes and change the name as whatever you want to. So this is just the name of the actual, the sequence file that you had without any extension. So that becomes a little easy for you to follow up later what is being done. So, and it is always good to give descriptive names because let's say you go back to the pipeline 1 year later, you may not remember what you have exactly done. So descriptive names will help you identify what exactly you have done here, right? So this is what you do. Then you click on save finally. So this is saved and your file name is now DRR187567, whatever is the number here, and it is tagged as uncompressed. You should use a hashtag here so that whenever you use this file, that hashtag will automatically carry forward and get tagged. And if you want to look at a slight view of your file, you can have it here as well, right? When you click on this panel, this will elaborate and then you can have a look. And then of course, if you want to look at the data in the central panel, you click on the eye. And now you see this is your Fastq file again. And this is now uncompressed, right? Although you don't see it's uncompressed here, but we know that this is uncompressed. Now we move on to the first part of our analysis. First part is to filter the data, right? Because you see the first read here is 218 bases, the second read is 382 bases, and then if you see the fourth read is 1285 bases, then this one is 561, 839, then you have 1668. So what you want to do is to filter first for at least, you don't want to include reads that are less than 1000 bases. So what you're doing here is you use filt long to filter out, and here in this case, you go to your tool shed, you search for filt long. When you click on this filt long, it will come in the central panel. It will ask you for files, so we'll upload the file that we have taken here, right? And sometimes it will automatically pick up the file. Sometimes you may have to do a bit of process, management to tell it what file you want, what file it is likely to take. So, and this, if you see here, you have a folder icon here, you have a file icon here, and you have multiple files here. So for now, we have only one file, so we'll just upload one file. But in case you had multiple files, the same process will be applied on multiple files together, right? And we'll come to that when we talk of nanoplot, where we'll do a nanoplot for both unfiltered and filtered data. So here you are. So you can, these are the options. We'll not change too much of an option, except that we are going to do a read manipulation. So here is your read manipulation option. And in read manipulation option, we are ensuring that the minimum length that we are taking forward is 1000. Anything less than 1000 is being discarded, right? So this is output threshold we're looking at, and we're looking at minimum length, and we're discarding anything less than 1000. Everybody here, class? You're following?
Yes, sir.
Sir, can we provide
Yes,
paired
sir.
and reads
Yes,
also?
sir.
Sorry? Can we provide paired and reads also? Like you have
Yes.
uploaded.
So if you have paired and reads, you'll have to specify that you have paired and reads, and then you'll have to have, and when you do with paired and reads, you have basically two different files. One is basically, mentioned as _1 for the forward read, and the other can be mentioned as _2 for the reverse read, right? And then of course you pair them together. So there is a process of pairing up files together so that the two files move together. But that is a bit complicated. We'll not go there for now. But yes, I mean, you can look at other examples. You'll be able to see that how to upload a paired and read. These anyways would come as two separate files, but you can link them together saying that these two files are from the same fragment. So that then the analysis also gets different. I told you, you can then look for indels and other things because you know what is the fragment length that you have. and what is the fragment length that is coming after you align to a reference, right? And if there is a difference between the two, you know there is an indel there. Remember, we talked of this yesterday.
We can upload, likewise, you're doing, likewise, we can do that.
Yeah, so
also.
then in that case, you'll have to click on this option where you'll upload 2 files, right?
Okay.
And then, so I'm on PowerPoint, so you'll click here, upload 2 files. One
Here
will
it
be forward, the other will be reverse, right? And then take it forward, right? Okay.
So can you please repeat the filter step again?
Yeah, so the
What
filter
option
step,
we have to choose for filtering our?
so here you're looking at these are the options that you can have. Output threshold is what we're choosing, right? Because we are selecting for only the reads that are greater than 1000 bases, right? So what we say is in the output threshold, we say, so this is when you click on output threshold, these options would open up. And here, if you see, there is an option for minimum length and there's an option for maximum length. We are not worried about maximum length for now. What we want is at least the read length that we take forward from here should be a minimum of 1000 and not less than that, right? So that is what we filter for because we are using long reads. Okay, clear? You follow?
Yes, sir.
Yes, sir. Okay.
Okay, so then we move on and we talk more about this. So then you click on execute and that will be executed. And then of course, if you see here, it will show up here as your third step, filter long on data 2. So this is your data 2 here. You can see this number. So this is your data 2 and you're filtering on that data and you have the filtered Fastq, right? So then of course, if you move forward now, you will have, filtered. So now, of course, you click on this, you're clicking on attributes here. So now once the process is done, you click on attributes here and you look at your file here. You can rename it a little bit. So you can rename it as filtered data. So that is what you're going to do here. So if you see here, so now we have renamed it as underscore filtered. And now if you see your first read is 1285. Earlier it was less than 1000 if you remember, right? So now every read is greater than 1000 because we have filtered it for 1000 minimum length. So any read that was less than 1000 is not included in our data now, right? So this is what we have done now. This is your filtered file. Now what we're going to do is we're going to plot for both the filtered and unfiltered data to see what is the level of elimination that has happened and what is the values that we get after filtering, right? So for that, what we use is nanoplot, right? So filter long we have done, and you could also look at, so you could click here for other properties. So there are, you can view from here, and then for other properties, you can view from here as well. You could visualize the data from here. This is the visualization button, and there are other things that you can do here. You can toggle and cross-check for yourself. And then, of course, now what we do is we plot the values that you've got from filtering and from the unfiltered data. So here you're going to use nanoplot, plotting suite for Oxford nanopore sequencing data and alignments, right? So this is basically a plotting suite. And here you select for multiple file mode. So that is your batch mode because you're going to give it 2 files. One is your filtered and unfiltered. And then of course you can give more options. So if you go here, so if you see, I have selected for the second one. I'm giving 2 files now. One is the filtered file, the other is the unfiltered file, right? So 2 files are being given here and both will be processed with the same steps and both will then give you equal number of outputs. So I think there are some 6 files from each of these, for the 2 files, it will be 12 files finally that will be formed. So this is where somebody got stuck in the initial part of the talk. Somebody was saying that I didn't get enough
No,
files.
sir, I got the plots actually. After
Uh-huh.
that only I was not, after assembly step, I got stuck up actually. I got
Assembly.
all the plots.
Okay, so then it is fine. So assembly is I told you yesterday also, assembly is the most intensive part of this entire exercise because you have so many reads and each read has to be aligned on. I mean, here you're doing a de novo assembly because they're long reads. So you have what you're checking for is overlaps between reads. And if there is a unique overlap, you're putting them together. So that is something. So this is like all versus all comparisons. So therefore, it takes a long time, right? So that is why command line is advantageous. If you know programming, that gets a lot easier. And if you have a high speed system for in your lab, then it becomes high performance computing. Then it becomes a lot easier to do it. But Galaxy is an alternate that can still get you there, but it will take some time, right? So that is what it is. So here we have selected two files, right? And we are comparing for the data in the filtered, where we have eliminated anything less than 1000. And the second one where you have all the data, including the reads that were less than 1000, right? And then of course you select for batch, then you select two files here by clicking on this multiple files option. And then of course you click on execute, and now your job is queued. So if you see there are so many files that will be, oh, I'm sorry, I'm in the PowerPoint, I tend to forget. So there are so many files that will be formed now, right? And you can see the tag also, the filtered files are showing up here and the unfiltered are below this, right? And then of course, once you're done with this, so you are basically, you can now view your files. And if you want to compare the two, you could basically use on the top of this, there is something called a scratch, right? And this one here is called the scratch. When you click on this, this activates and you can now open two or more files together in live mode and compare them together. For example, here I've opened the summary statistics file for data 3 and data 2. Data 2 is unfiltered, data 3 is filtered, right? And if you see here very clearly, the mean read length here is 872.1 because we have filtered out the smaller reads. In this case, data 2, the mean read length is lower because the smaller reads are also there, right? And you know that mean is strongly affected by larger values than smaller values, right? Then, of course, mean read quality is almost the same for both. Median of read length is, again, quite large for the filtered data as compared to what it is for the non-filtered data. So I have another slide where you have more clear comparisons. Then you have this one here. So this is your, again, the plot we are comparing. This is log transformed histogram read length. And you can see we have filtered out anything less than 1000. So there's nothing less than 1000 here. And in this case, we have reads up to, this, if this is 100 and this is 1000, so this would be around 200, 250 and so on and so forth. So you can see that very clearly the graph behaves differently, and the N50 values also change slightly with respect to this, right? Because when you eliminate the shorter reads, the N50 value goes towards the higher side now. So here you are, if you do a comparison here, so this is your not filtered, this is your filtered. So mean read length here for not filtered is much lower as compared to filtered because you have removed the smaller read lengths. Mean read quality is approximately the same here. Median read length is again higher for filtered, less for unfiltered. Median read quality is also higher for filtered, less for unfiltered as expected. Number of reads, 91,288 is your original number of reads, and here it is 69,906. So you have effectively removed around 20,000 reads here, right? You understand that, class? So roughly your 20,000 reads were in the range of less than 1,000. So those have been eliminated in your filtered data. Then read length N50 also would increase here because you have already removed your shorter reads, so 15,102. And then, of course, total basis. This is again, this will be much lower here because you have eliminated a lot of reads here. 20,000 reads have been eliminated. And this is a lot more here because this is the entire data here. So around 2% difference comes with respect to total number of bases you're looking at. You follow, class, everyone?
Yes, sir.
Class?
Yes, sir. Yes, sir.
Okay. So then we move on. So this is your comparative summary. Now we come to the next step, that is your assembling the long reads. And for assembling the long reads, we're going to use Flye, and we are going to do the assembly only with the filtered data because we know now that the filtered data is much higher quality as compared to the unfiltered data. So again, we search for Flye here, and then you click on this. Flye would come in the central panel. The first option, of course, always would be input reads, and then you input your file here. This is your filtered data here. Nanopore raw, and you don't play too much with the options that are there. You just go to your filtered file here, and then of course, once you do this and click on execute, this will come finally here, and this is now finally your the output of Flye we are looking at, right? So if you see here, Flye on data 3, and we're looking at the consensus here. So this is your actual contigs here. And there are two contigs here, but because this is too large, it is not showing the other one. But you can click on other files that will give you that. So for example, you could look at assembly info, right? So if you click there, you see this is assembly info. You can see very clearly there are two contigs, contig_1, which is the larger one. And this, is now your bacterial chromosome. And this one, contig 2, this is the smaller one, 60,128 bases. This is likely to be your plasmid, right? In comparison, you can see that this is going to be very large, this is going to be very small. So, and we know in Staphylococcus aureus, there is one plasmid and there is one this thing, right? And then, of course, if you look at the coverage and everything, so you have the coverage of 171, the 755, circular, both are circular. Repeats are not present in the chromosomal DNA. It is present in contig 2 to some extent, and so on and so forth. So other properties are mentioned. Then you move on and you go to Bandage to see how do they look graphically. So you click on Bandage and you again give the inputs as the final consensus data, and that would basically, give you the images. So you can very clearly see there is one large DNA. So this is proportional to the size that is there in the contig. And there is one very small DNA here. So this is going to be your chromosomal DNA. This is going to be your plasmid DNA here, right? So this is how it goes on. Then you move on and we can apply now to check for the quality of the contigs. You can apply what is known as a Quast. So again, you search for Quast here, and then you click on Quast here, and that would be basically apply your files here again. So Quast on data 14, right? So this is your, and this will generate a PDF and an HTML report of the, and this will again generate some graphs that will tell you what is the quality of the assembly that you have created using Flye, right? So here you are again. So this is Quast report. This will be in multiple pages. It will give you the idea of how good your quality of assembly has been. And then, of course, you can move on and you can use, you can now search for your genes that are responsible for your, whether your assembly has genes which are known to play a role in AMR or antimicrobial resistance. So for that, in this pipeline, I've used Abricate. In the original pipeline, they've used star AMR, if you see. But for some reason, star AMR is not working. At least for the day I was working on, it was not working. So I've used Abricate for the purpose, right? So Abricate also is a tool that can tell you, that can tell you, about the genes that are playing a role in antimicrobial resistance. To screen for, so if you see here, this is your Abricate mass screening of contigs for antimicrobial and virulence genes, right? And this is the one-line annotation for Abricate, how it works out. And then, of course, you can give your Flye data, Flye. So this is basically your assembly data, the consensus data that you had, where you had the sequences of both the contigs. And then, of course, you can click on execute. And once you click on execute, it will work out and give you the output files here. So this will be your, Quast on data. There'll again be an HTML file. So this is a Quast. We are now talking of Abricate. So let's go to Abricate now. So once the job is done and Abricate reports are there, you will have the report file here. So Abricate on data 14 report file, and then this, when you click on this, you'll be able to see whether it contains genes which are already reported for playing a role in antimicrobial resistance or not, right? So here you are. You can see there is an ANT9 gene, LA_1. There is an ERM gene. There is a TET-M gene. And then you have again ERM, and then you have the MEC-A gene, AAD gene, and so on and so forth. So these are known to be players of antimicrobial resistance. So now you know that the bacteria that you're looking at is actually methicillin resistant, right? So it is basically a resistant form of bacteria that you're looking at. Without assembly, you would not have been able to make out whether it is resistant or not, right? So this is the function of Abricate. And then we come to the last tool that is going to be Procca, right? So if you go to Procca here, so next we move to Procca. Yeah, so this is Procca, and Procca again, you can give your assembly and then of course, and then Procca will give you a detailed annotation of your of the contig that you put in here. So here you are, it will give you from position X to Y, what is the kind of, the gene or what is the kind of CDAs, so on and so forth. So here you are, if you see, you start from 109 to 1655. There is a ribosomal RNA. This is on the plus strand. And then you have additional details here. Then you have ribosomal RNA from 2127 to 5040, again on the plus strand. Then you have the third ribosomal RNA again on the plus strand. You will have a score also to indicate how confident you are of this. Then you have CDS, and some of them are on the plus strand, some of them on the minus strand, and so on and so forth. So this is your detailed annotation of your actual genome that you've assembled now, and you're looking for this, right? So this is how this is done. And then, of course, Galaxy can be used to extract the workflow. So you can go to your history here. Let me just pull this down here. So you can extract the workflow, and that workflow can then be used again as and when you want to use it for your analysis again. And you can just start feeding your data here. And once you can click on create workflow. This will create a workflow now. So this is your workflow now. And this workflow can be used for a fresh data that you want to use now, right? So this is the advantage with Galaxy, that you can use the workflows again and again. You can change the type of, so let's say you have another set of data coming from another site. So you can just feed the data on the top. You already have your pipeline ready, what tools to use, what is the type of input that is to be given. And you know that here, the output of one process is the input for the next process. So the files are created accordingly. So exactly the same process will be followed, and very smoothly your process will be done until the end. So this is the advantage that you have with Galaxy. And likewise, when you do hard coding, we do it this way again. We make pipelines so that the output of one is the input for the other. You may have to change formats a little bit here and there. So those are special wrappers that we write to ensure that, the file formats change and it is easy. So this is where we are. We are done with this for now. In case you want some books on R, you can have a look here. So these are all available online. A little book of R is available online. The last time when I took this workshop, somebody asked for books on R. So a little book of R is available. Bioconvector tutorial is available. Biostring quick overview is also available. Introduction to Bioinformatics with R, a practical guide for biologists, is also available online. And then Omics Association studies with R and Bioconnector are also available. So you can have a look at that. Now I'll give you some practical demonstration of how you go about doing things. So we are done with this part, but let me just take you to Galaxy again and give you some practical demonstration, right? So here we are, right? So this is what we have. So I was redoing the entire thing again, and if you see here, this is how it is. So Flye assembly info, right? So this is, I could reach only the Flye step till now, because somehow the server is taking very long to process, right? So what you can also do is, if in case you go wrong, you can delete your files from here and redo the step with correct inputs again, right? So for somebody who got stuck, you could delete your process here. When you delete these files, your process would start from the next number. So if you delete the 20 file, the next process would be 21 here. It would not be the 19 to 20. So you'll see there is some discontinuity of numbers that happens every time you do your here. So for example, here, this is your first step where I uploaded the data, and then you see there is 2, 3, 4 are missing. So that is where I did some errors, and I had to delete those files. And then finally I came back to step 5, which is actually step 2. So this is one thing. Then whatever tool you want to search for, you can just click here. You say filt long, let's say, right? And you see it is coming up here, filt long. And you click on this. Now it will come to the central panel. And once it comes in the central panel, you can, give your data here, and this one here. If it is single file, you can give a single data set. Multiple data set, you can give multiple files. So if you want to give a complete folder data set collection, you can do that as well, right? And then of course you can add up here, and then you could do your the thresholds you can set. Most of the thresholds you can go by default, because they are mostly set for specific type of analysis. But in case and you very specifically know that you need to toggle with certain positions, you can do that. For example, here we had toggled with output threshold. So we had set minimum length of 1000, right? So I'm not putting up a file here for now, but just to show you, this is how you can do. And then you can click on execute, and that process begins, right? So if you see here, Assembly info on Flye. So if you remember, we had got 2 contigs. So if you click on display here, you should be able to see the assembly info on. So here you are. This is your contig 1. The larger contig, the coverage is 171, circular, repeats are none, and so on and so forth. And then you have contig 2, which is this, right? And then if you want to edit the attributes here, you can click on pencil here. You could change this name here. Most commonly, you'll change the name. Sometimes you may have to convert the files, right? So if you see here, if you convert the files here, you could have some options, but not depending on which step you are, you'll have different options, right? Then you could also look at the data type and permissions. Generally, unless if you're registered, your email would be active and you would be allowed to do most of the things. Then if, let me just show you here, for example, if you were in this data here, right? And you look at this, you click on the edit button here. And let's say you want to convert this. So this is Fastq BZ2 and you want to convert it to uncompressed format, right? So here you are, you could then, Fastq Sanger to Fastq. So this is the format here. So this is .bz2 to Fastq Sanger, which means the extension is gone, which means it is uncompressed. You can select this and create data set. It will create a data set for you, right? Then, of course, if you want to move on forward, let's say you want to, from Flye, what did we do? We did the Bandage, right? So we can click on Bandage here. Let's see if we can do one live exercise. BANTAGE. Class, you're following everyone?
Yes, sir.
Yes, sir.
Yes, sir.
So we go to Bandage image, visualize de novo assembly graphs, right? So this is the function of this. So we go here and here we give the assembly info, right? So this is the assembly info that we have, and we'll give this, and that should basically work on it. Let me see. Assembly info, or I think it should work on assembly. Yes, assembly info should be fine because it doesn't really require the sequence to get done, right? So that should work. We'll see, right? And let me just cross check what we'd given here. I don't want to go wrong here, and almost it is always a bit of trial and error when you go about doing this, right? So this is basically, if you see here, this is on assembly graph image, right? So this is Bandage image on data 16. What is data 16? Graphical fragment assembly, right? So this is what we need to give it here, right? Flye on data 3. Graphical fragment assembly, right? So this is what we're giving 60, right? So this is what we'll give, graphical fragment assembly. So here you are, you go back here. Let me see, you have this. Yeah, this one, right? So this is the input file for this. So this is something that as you use the tool, you'll get to know, right? Everybody, you follow? And let's see if this works out.
Yes, sir.
Keep everything fine and let's see if this works out. So we say execute. Sometimes you know what is Murphy's law? Anyone? Murphy's law? Hey, Murphy's law nahi pata. Murphy's law is basically things that you would least expect would happen at the wrong times. So for example, you're going late to your office. That is the only day when you'll get your crossing closed. So you'll be further delayed. So sometimes it so happens when you're going to display something, that is the time when it doesn't work. You're telling the class that you'll get a red color after the final adding of solution, and finally what you get is a blue color. So that is something that is Murphy's law. So here you are. This is your data here, and you can have a look at this. Here you are. So this is perfectly fine. No Murphy factor here. You have one assembly here. You have the other assembly here. So if you remember, there was one chromosomal assembly, and the other was the plasmid here, right? Everybody, you follow?
Yes, sir.
Yes, sir.
Yeah, so this is how it works out. So in the central panel, you'll be able to see this. And then, of course, you can go forward from here and you can use Quast. So let's do the Quast thing and let's see if that works out. Fortunately, it is working quick here, although the Bandage thing is not so intensive. So QUAST is what we're going to use here. And you have the Quast genome assembly quality. And this will be on Flye Data consensus. It has automatically picked up the file that it's going to work on. So then you can click again on execute. We are not toggling too much with any of these values, default values we are working on. So we say click execute. And now again, if you see, this is for now gray, which means it is queued now. Then it will turn orange, which is basically orange or kind of beige, which will be basically to show that it is now processing. And then finally, it will be green when the process is done, right? So that is how it works out.
Excuse me, sir.
Yes.
Can it draw a circos also?
Circos, you will have to see, right? So let me see if Circos is here. I'm sure it must be there, but Circos is there. Yes. So you can have Circos. Can you see that?
Yes,
Yeah.
yes. Okay.
So it can do a Circos. So basically, most of the tools that are there are now populated here. So that is the advantage that you have. But the only disadvantage with Galaxy is it is going to take time, right? And we are still doing on a very small set of data. If you have a very large set of data, it is going to be, sometimes it may be very exhaustive and it may not work out. So therefore, so let's come to what you require if you want to be a data scientist and if you want to be in genomics. So first thing important is you need to have a bit of knowledge of programming. So you must know a bit of Python. You must know a bit of R, right? That is important. You can go to my my YouTube channel that is called Dr. Vipin's Biotechnology, Dr. Vipin's Biotech and Bioinformatics Classroom. The logo that I use in my PowerPoint here, right? So if you see here, not this one here, right? So if I were to reduce this a bit. Okay, so this is not the one. Let me take you to other slides, beginning slides. So here you are. Okay, because these are overlapping with, so this is the name of the channel, the YouTube channel. I have both tutorials on R and also on Python and also on Linux command line. So the three things that you need to know, apart from, while Galaxy is good, you should also have, I mean, because your data will be huge and you will be working on very extensively. So it is always good to have control on what you're coding, right? So therefore, you must know a bit of coding. Python, R are the industry standards. And then, of course, Galaxy is help. So these are three things that you need to know. And of course, Linux environment is something that is, again, important. So if you have a combination of these three, that helps. So that is something that you need to know. And also, of course, you need to know your sequencing platform that you're using and its nitty-gritties is something that you need to know very clearly. So the more you read, the less time you will spend on the bench. The less you read, the more you be on the bench and you keep working and you don't really get to good results. So the important point here for a researcher is reading is to be the first important thing. You have to read a lot, right? And reading has to be extensive, not just confined to your subject, because everything is interlinked. We're talking of systems biology, so we're not a gene is not functioning in isolation, a protein is not functioning in isolation. It is working as a network and together. So therefore, the more you know, the more you'll be able to link things, and the more you'll be able to produce good quality papers. So that is where we are now, right? So let us see if our process is finished, and let me see. So it is still queued. So it is not even begun now, right?
Thank you, sir.
Yeah.
Sir, would you provide your email ID or some contact access?
Yes. So my email ID I'll share in my, so probably the presentation is over, so I'll stop sharing. And we still have 4 minutes to go. Any questions I'm willing to answer. And let me give my email ID in chat so that you can have, So this is vipin.singh.cu@gmail.com, right? So here you are. You can have my email ID, and in case you have a query, I would be happy to answer in case I'm able to answer. That's the only thing. And in case.
Thank you so much, sir, for such a wonderful class, for such a nice class.
You're all welcome, class. I see there is a lot less attendance. I think it is towards the end of the 1 month, so probably people have moved on.
I
Right?
have a doubt. How do you go for examples for practicing this protocol?
Sorry?
The examples, how can you search for papers? for trying put as examples.
Yeah, so that is again, you like the paper that I had shown, that would be one starting point, right? Then Galaxy has a lot of tutorials. I had shared a link with you. Other than that, you can, just take any paper and go back to its data and start analyzing using Galaxy. I mean, it is again, the more you read, the more data you will get, the more you will be able to, the idea is, so as I said, and when we are doing R also, I said, real learning will happen when you have a real problem in hand, right? So either you take somebody else's solved problem and do it yourself, or when you are directly into a project, when it is your data and it is your personal data, as in the sense that your own PhD data or a project data, then of course it is your responsibility to do it. So then of course you start finding ways of analyzing it better. So that is how it happens. But for practice sake, you can just go to Galaxy Network. There is a lot of help that is available, and a lot like the file that I had shared with you. Each step is mentioned, how to go about it, what is the input to be given, what is the output to be expected, what are the questions each step is answering. So this is how it works. All of you, thank you very much for your comments, and it is very, very well.